YouTube channel. And Ibrahim has said he's happy to take questions throughout the talk as well as at the end. So if you do have burning questions throughout, feel free to either unmute yourselves and ask, raise your hand, or if you'd prefer to type in the chat, I can read the questions out as well. So thanks so much, Ibrahim, and please take it away.
Thank you very much for the introduction. It's also very nice to be here. Today, I will be talking about how we use deep learning models to design, first to understand the code of enhancers and to design cell type specific enhancers. You hear my screen and a mouse, right? Yes. And
yes, perfect.
Enhancers, they are the core elements of genetic networks where transcription factors, they recognize short motifs on the DNA sequence and they bind. And their combinatorial and differential expression of these transcription factors, they orchestrate cell type specific gene expression. And during my PhD, we were studying the code of enhancers in two main different systems, Melanoma and Drosophila brain. And we were training deep learning models on DNA sequence to understand the code of enhancers. And in this recent study, and today I will present, by using those deep learning models, we were trying to design cell type specific synthetic enhancers. I will start with the melanoma part. Here, very brief summary for the melanoma. Here we are working with the cell lines, melanoma cell lines. There are two main states of these cell lines, melanocyte-like state and mesenchymal-like state, proliferative and invasive. I will mention as mel and mes. In one case, SOX10 and MITF transcription factors are highly expressed, and for the mes is AP1 and TED. And to start with, to get the candidate enhancers to study, we performed ATAC-seq on 17 patient-derived human melanoma cell lines. As you can see, the ATAC-seq profile for two different states. Some peaks are generally accessible, some are cell type specific, some are state specific. And to get the state specific and cell type specific enhancers, we ran cystopic and we clustered co-accessible regions. And here, for example, there are 24 topics. In each topic, there are around 3,000 regions. For example, for topic 1, these are the ones that are accessible in all the cells. And for the topic 4 and 7, as you can see here as an example, these are the classes that we were mostly interested for the cell type specific accessible regions. These are our candidate enhancers. And by using this clustering results, we trained our deep learning models on these DNA sequences. So our models, they take 500 base pair DNA sequence as input, and it tries to predict in which topic or our cluster cell type that piece of sequence will be accessible. This is our model, and then we train, then we validate, we check the performance is all good. And then we are mainly interested in not like the performance of the model, but what the model learns and how can it correctly identify cell type specific accessible regions and then enhancers. So we were using certain techniques to understand what model sees on the DNA sequence. So I will give an example how we do. So here you see the IRF4 locus, where its enhancer is on the 4th intron, and it is specifically accessible in the MEL state, but not in the MES state. And by using the MEL state specific class topic, we score, okay, our model takes 500 base pair DNA sequence as input, but we can score all these 500 base pair pieces with a sliding window. And the model says, okay, the enhancers should be here. Then we ask the model by using network explaining techniques, like how can you know the enhancer is here? And it tells us it's because of these nucleotides. So like, okay, now here is the 500 base pair, but you see the ones that are more highlighted based on the model, thanks to these deep network explaining techniques. And these are the nucleotides used by the model to make a correct prediction. And if I zoom into this 200 base pair space, now you can see more clearly the transcription factor binding site patterns on this 500 base pair sequence. And some of them are positively contributing, we call activators, and some are negatively contributing, we call repressors. And with this way, we were able to see, okay, which transcription factors are binding on an enhancer or the DNA sequence, positively or negatively. And there's another way to see like what model see on the DNA sequence to make a correct prediction, which you can we can understand is by using perturbation experiments in silico. So here I show a saturation mutagenesis experiment where we do Okay, here we have A, if you mutate A to other 3 nucleotides one by one, and how the prediction score is changing compared to the wild type original sequence. And when we do for all these individual nucleotides one by one and then plot all the results, it's like called saturation mutagenesis, and we do this in silico. And with this enhancer, we were lucky because like another group, they did the same experiment in vitro. They mutated each single nucleotide one by one and then measured in vitro activity. And they calculated log2 fold change compared to wild type. And as you can see, our in silico single nucleotide predictions, it was correlating quite well with the in vitro experiments. And we actually, just from this single enhancer and with the help of this in silico and in vitro experiments, we learned a lot from this enhancer. As you can see, if you do mutations on the activators, the activity of the enhancer is going down. And also there are other type of mutations, as you can see, these like individual ones, where it doesn't correspond to any motif. And actually, these are the ones that create repressor binding sites. So after, for example, this mutation, like this is the wild type, and if I apply this to this A to G mutation, it creates now a repressor binding site, which decreases the prediction score of the model, and also it decreases the activity of the enhancer. And there are, on genomic enhancers, there are many of these, like in IRF4, there are maybe
Okay. Hi. Did everyone lose Ibrahim's?
Okay.
Yeah.
Okay. Great. Not great. I mean, I will try and get in touch with him, email him, and hopefully his Wi-Fi will recover.
okay. So it was me who dropped.
Yes.
Yes.
Hi, Ibrahim. Great to have you back.
Yes.
Yeah,
was I gone too long? I noticed at some point, okay, I don't see myself anymore. So
yes, you were out for maybe like just over a minute.
okay. So from this slide.
We got the introduction of the mutation for the ZEB motif.
Okay, perfect.
Yeah.
Okay.
I know I'm not showing, sorry. Yes.
Yes.
This was the slide, I think. Okay,
exactly.
perfect.
Yes.
And yeah, I continue. So this was the wild type, and with these single individual mutations, they create repressor binding sites. And as you can see, after the mutation, it creates a repressor binding site. And I was saying that on genomic enhancers, we saw this, like there are many these single mutations that create repressor binding site. And on IRF4, maybe it is 10, 15. And I can show like another examples. And here, this one was interesting one because like it's a common SNP in European population. It's a C2T mutation. It creates a repressor binding site, and actually it's associated with a phenotype. If you have this mutation, you have brighter hair or eye colors, and actually it's like it is not IRF4. This is the enhancer of IRF4. It's transcribes transcription factor, and this transcription factor is non-regulate tyrosinase, which regulates melanin production, and a single mutation on its enhancer of this IRF4 transcription factor decreases activity, which indirectly regulates the phenotype of a person. And okay, to decrease the activity of the enhancers, you destroy the activators or create repressors, and the other way around, to increase the activity, logically you destroy the repressors or create activators. And also what you can do is like you can increase the affinity of already existing activator binding sites. For example, here the MITF, it's not its perfect binding site. And with this mutation, it's the affinity increases, the pattern is now better, and it increased the prediction score and activity of the enhancer. as in this example, it creates a better SOX10 binding site, and it increases the activity of the enhancer. So inspired from this picture, and also we were studying cross-species enhancer code, we want to see if we can increase the activity of enhancers that are already active in another species. Here we took a sequence from a dog, and as you can see the binding sites here, and the model says this mutation and this mutation, they are the ones that increase the prediction score a lot. And when we apply this mutation, it creates a better SOX10 binding site. And the other one is like destroying the repressor binding site and creating the activator at the same time, because between MITF and ZEB, they are both like ebox like motifs, and the only difference is like for the MITF the middle is CG, and for ZEB middle is GG. And if we change this G to C, we saw that by just 2 mutations we were able to increase the activity of an enhancer by 10-fold. And then similar to this, now we took orthologous enhancers in between 2 species, but this time one of them is accessible and active in one species, but in the other one it is not accessible nor active. And if you look at the explaining which motifs are present, the model says to us that in the human one, the core transcription factor binding sites are destroyed maybe during evolution. Maybe it's an enhancer that was lost during evolution. And if we compare these 2 500 base pair DNA sequences, there are actually maybe 100 mutations. But with the help of model, we can choose the most crucial 4 mutations. And when we apply those mutations, we were able to rescue the activity of an enhancer that was probably lost during evolution. Then, okay, we can enhance an already active enhancer, and we can rescue an enhancer that was lost. And then we ask, like, can we create an enhancer from scratch by using this approach? And here our design process, it's very straightforward. We start from a random sequence. Random, I mean not from a genome, but it's like completely random by using NumPy random function. And then we calculate saturation mutagens on this random sequence. And when we choose the mutation that increases the prediction score highest, and then we apply this mutation, and then we do this procedure iteratively. After applying the mutation, we calculate saturation mutagens again on the whole sequence, and then we choose the next best mutation, and then we apply. And here you can see initial random sequences, the prediction score is almost 0. And with the number of mutations, as you can see, how the prediction score is increasing for 4,000 random sequences. And to test this, we selected 10 of them at 15 mutation. And here you can see the luciferase results. The red ones represents the genomic enhancers we used as positive control, and the green ones are the ones that are evolved from scratch by using saturation mutagenesis. And as you can see, the enhancers that were created from scratch, they are distributed quite well in terms of their activity levels compared to genomic enhancers. And if you look at how it looks like the random sequence and after 15 mutations, as you can see, there's no activators and repressor. There are repressor binding sites, and after 15 mutations, this repressor binding site is destroyed and more activator binding sites are created. And since we do these mutations one by one, actually we can see all this trajectory, what each mutation does. For example, first 3 mutations, they create a very strong SOX10 binding site, and in the meantime, actually we can measure the activity. The random sequence, it's not active. With a very nice single SOX10 binding site, it's not active. Then on the 4th mutation, it converts the repressor to an activator. It's, I think, still not active, maybe slightly it's increasing. But when more cofactors or like the other binding sites appear, like here's TFAP or a weak SOX10, then the enhancer becomes active. And when you have an active enhancer here on this step, we create the same SOX10, it creates the same SOX10 binding site as in the first one, very strong one. And when it's active, since it's past the threshold of the activation maybe, now it gives a very high boost in terms of enhancer activity. And with this way, we were able to track what is the effect of each single mutation on the enhancer during the design process. And as I was mentioning, the repressors, okay, we observed them on genomic sequences and on the random sequences. And then we thought, okay, we wanted to do more experiments on these. I'm showing the same sequence as here. So initial random sequence after 15 mutations. Now we choose the mutation, the nucleotides, as I was showing on IRA4, the single mutations that creates repressor binding sites. But we choose the mutations in a way that they do not disturb the activator binding sites. It's in between or in the flankings, as you can see. And the model prediction says, okay, after creating these 6-7 repressors, the prediction score drops dramatically. And when we test this in vitro, as you can see, even though we didn't touch any activator binding sites that are important for the activation, and creating this repressor binding sites in between, or at the flankings, it destroyed the activity of the enhancer. Then we ask, okay, what if we create this repressor binding sites one by one? And here you can see, like, without any repressors and having only one, then we saw that the more you add repressors, the more the enhancer becomes less active. And after 6 of them, then it becomes closed. The enhancer is not active anymore. So it means, like, it gives you another kind of possibility to play, not with the activators, but with the help of repressors, you can play more with the activity level of the enhancer. In terms of what you like to have to have a certain level of activity. So then, okay, so far, like all the enhancers that we design, we are testing them in vitro, in this human case, at least in vitro, with the luciferase assay. Then we were wondering what would happen if they were integrated into genome, and how would the accessibility profile would have affected, or the gene expression, or histone acetylation, and so on. So for this purpose, we used Enformer. Enformer is a deep learning from DeepMind, which takes 200 KB enhancer or DNA sequence as input, and it predicts all encode tracks. Like there are maybe 6,000 tracks from CAGE to ATAC to DHS to chip transcription factor, chip histone modifications. And for us, SKML is also a melanoma cell line, so we were using the SKML5 to have the correct approximation. And as you can see, we put the IRA4 locus as an input, and this is the random sequence profile. And we put our synthetic sequence where the IRA4 original enhancer is. And as you can see, the more mutation happens during the design process, the region becomes more accessible. This is DNAs. And when we introduce repressors, and the model says, okay, it decreases the accessibility as well. And on Enformer model, there are, for the DNAs, there are maybe around 600 DNA tracks from different cell lines and tissues. And if we check after the 15 mutations, which tracks have the highest prediction score among the 600 different cell lines and tissues. And the top 4 are like 2 of them are SKML, 2 of them other type of melanoma cell lines. So it shows us that it's very specific to the enhancers that we designed with the help of our model. Now we check with another model, completely independent, and different inputs and outputs. And it shows that, like, the enhancers we generated are specific to melanoma and melanocytes. And then we did this experiment now in vitro. We integrated our cassettes where there's enhancer, minimal promoter, and the luciferase gene. As you can see, like, this is the cassette we integrated. Then we performed ATAC-seq on these cells. And if you look at our cassette and the accessibility profile, the random sequence, it doesn't have accessibility. And after 15 mutations, it becomes accessible. And when we introduce the repressors, and the accessibility has again decreased. And as I was saying before, the Enformer has also the other type of tracks for gene expression and histone modification. And if you look at the CAGE, and again, we found another cell line that is close to the cell lines that we are using. And as you can see, from random to evolved sequence, it also increased the gene expression prediction by using the CAGE, and repressors decreased the gene expression again. And for the histone acetylation, here we use H3K27 acetylation for the foreskin melanocyte. And again, our enhancer, the designed enhancer, is also able to drive histone acetylation. Okay, so far all the things I was showing is in vitro and using the cell lines, and then we wanted to go further, and for this purpose we start using our Drosophila brain model from another publication, and we have already trained the deep learning model also here. Then we start using to design synthetic enhancers. but I will give a brief introduction to this paper. Maybe you may already know. We perform single-cell attack-seq on a developing fly brain from larval stage until adult at different time points. And we had RNA-seq data already, and we combined for the adult brain. We combined with the attack-seq, we co-clustered them, and we transferred the cell line, the identity of the cells. And we also identified cell type specific accessible regions for different genes and for different cell types, as you can see. And finally, like we built enhancer mediated gene regulatory networks where, as you can see, there's here the transcription factors, the core transcription factor for gamma Kenyon cells, for example. They bind to these blue accessible regions and enhancers, and they regulate this outer reddish circles that represents the target genes. So these are our enhancer-mediated gene regulatory networks. And this part, I was mostly involved in the deep learning part. And for our models to train, we further clustered. We chose the glial cells, Kenyon cells, and T neurons, and further clustered them. There are like 15, 16 cell types. And we identified, again, similar to melanoma, cell type-specific accessible regions, or state-specific accessible regions. Here you can see these are like glial-specific accessible regions. These are neuronal accessible regions, and there's like Kenyon cells, three different type of Kenyon cells, and so on. And when we have these regions, then we trained our deep learning models. Again, as the same, it takes DNA sequence as input, and it tries to predict in which cell type this piece of DNA sequence will be accessible. And focusing on the Kenyon cells, with the help of network explaining techniques and TF-MODISCO, we identified that one-cut iris and MEF2 are the core activators for Kenyon cells, all these three subtypes. And more for the repressor patterns, we identified that there are more patterns that are specific to Kenyon cells, and even like some of the repressors are really subtype specific. For example, this 893F, the model says it negatively contributes in alpha beta Kenyon cells, not in gamma Kenyon cell, alpha prime beta prime Kenyon cell. And if you look at the gene expression of this factor, it is actually, indeed only expressed in alpha beta Kenyon cells. And that's why the model somehow knows. So we don't give any transcription factor expression data, we just only give the DNA sequence and corresponding accessibility on different cell types. But the model was able to learn which factors are contributing. I will give an example. So here is the SNPF gene, and its enhancer is accessible in 2 out of 3 Kenyon cells. And if you look at the expression of this gene, it's indeed expressed in those 2 Kenyon cells. So this might be the correct enhancer. And if we test the enhancer activity in vivo, where we have the enhancer minimal promoter and GFP, and it is integrated into the genome of the flies, and when they're adult, we check where the expression is, and it is as expected, the enhancer is expressed in the Kenyon cells. And if we score this enhancer with our model says, okay, it's the Kenyon cell enhancer and mainly these two subtypes. And now with the network explaining techniques, if we look at which nucleotides are used by the model to make a correct prediction, as you can see, these activators, one-cut, IDS, MEF2, and SR, it is all present in different subtypes. Okay, so what you see here is they are all same 500 base pair DNA sequence, but we are looking through different glasses because in different cell types, there are different transcription factor expressed, like different TF repertoires are present. So the model knows those intrinsically, and when we look through the eyes of the model with 3 different ways, we can see which patterns are more important for which cell type. So activators are all important for all 3 subtypes. But when we look at alpha prime, beta prime, there are these repressor binding sites close to activators, and they are negatively contributing in this cell type, and they are close to activators, and they are probably blocking the accessibility of this enhancer, as you can see here. That's why it's not accessible anymore, and its target gene is not expressed anymore. And as I was saying, like these patterns are also present in alpha beta and gamma Kenyon cells, but the transcription factor that binds to these patterns doesn't present in those 2 cell types. That's why the enhancer is functional in those 2 cell types. And focusing on these activators to see if they are real ones, what we did is we performed these 2 mutations to destroy MEF2 site, or these 2 mutations to destroy the IDS binding site. And each time we lost the activity of the enhancer. So it means like they were really the binding sites driving the activity of the enhancer. Okay, now we have another nice model. So by using this, we want to also try to do the same experiments as we were doing in human to design synthetic enhancers. And here you can see again initial random sequence with the number of mutations, how the prediction score evolves. And one interesting thing here we observed, after 10 mutation, if you look at the distance between, okay, the model does many mutations on a given sequence. If you look at the distance between the farthest mutations on a given single sequence, on average, it was around 200 base pair. Even though we gave the model 500 base pair, it could do any mutations anywhere it wants. But at the end of the day, it's mostly focused on 200 base pair space, which corresponds to a size of a nucleosome. And yeah, similarly, if you look at the random sequence, there are these repressor binding sites on the repressors on the random sequences, they are purely by chance. And we see them more often on random sequence compared to the activators because, as you may notice, they are usually short, like 4 or 5 base pairs. At least in the cell lines or the cell types that we studied, we always observed that these repressor binding sites are very short. And on a random sequence, it's easy to, by chance, they may appear by chance quite easily compared to the longer patterns, as you can see here is ILIS or MEF2. So, okay, in the beginning on the random sequence, there are repressors, and after 10 mutations, the model destroys the repressors and creates activators instead. And in this case, we focused on the Kenyon cells, and we tested 13 of synthetic ones, either with 10 mutations or 15 mutations. And 10 of them were positive and quite specific for the Kenyon cells as we were targeting, and 3 of them were negative. But we also observed that like we were co-staining with Dachshunds, which stains the Kenyon cell. And we observed that actually those Kenyon cells were not present or they were abrupted, which suggests maybe the GFP is too toxic and those enhancers were super active and they were during the development or at the adult stage, the cells were destroyed. That's why we were not seeing a GFP signal, but also the Kenyon cells were not there anymore. And okay, here, like with 15 mutations or 10 mutations, we were able to create an enhancer from scratch. But then we also wanted to see if we can manage this with less number of mutations. And for this purpose, we used the fly genome. What we did is we scored the whole fly genome, everything, not just accessible regions and so on. And we plot here the prediction scores versus the accessibility for the Kenyon cells. And here, this part, the model says, okay, it gives a high prediction score, but if you look at accessibility, they are not accessible. These are actually our model's false positives. Model says, okay, here's a high prediction score, but if you look at the accessibility, they are not accessible, so they are false. Is there a question? No.
No, I think it was someone who wasn't muted and had some background noise.
Okay. And what we did, we took these sequences and we introduced further mutations. Here we did just 6 mutation. And as you can see, like this is the wild type sequence that is present in the fly genome. And as you can see, ILIS1, CUT-SR, and weak MEF2, they already exist. But also at the flankings, as you can see, there are repressors. And when we choose the 6 best mutations with the help of model, as you can see, it destroys the repressor binding site. It creates another 2 weak ILIS binding site. And when we test this in vivo, here you can see the original false positive, and after 6 mutation, and they were becoming active enhancers, and we were calling them false positive or like near enhancer sequences that exist in the genome. Maybe they are like remnant of the evolution. They were enhancer before and they were lost, or maybe they are close to become one, and we are finding them by chance with the help of model. And another example I want to give is the repressor case, as I was showing in human genomic or synthetic enhancers. We want to test the same. And this is a synthetic enhancer after 10 mutations. And again, with single or double mutations, with the help of saturation pathogens assay, we choose these mutations and created repressor binding sites in between activators. And when we test these sequences, and every time, like we did 6 or 7 experiments from an active one and creating repressors. And each case, the activity of the enhancer was destroyed. And further, okay, I will give different applications of this way of studying enhancer code and synthetic enhancers. Here's another example is in this case, okay, like we start from a random sequence and we choose the mutations that increase the prediction score for the Kenyon cells, but actually our model is multi-label, multi-class model. So it knows not just the Kenyon cells, but it knows different glial subtypes and T-neuron subtypes. So, but so far the examples I was showing to create an enhancer from a random sequence, we were always choosing the mutations that increase the prediction score for the Kenyon cells. So here instead, starting from the random sequence, say the same random sequence, these are the same random sequence, but we are again looking through different classes for the Kenyon cells and for preneuronal glia. This time, if we choose the mutations that increase the preneuronal glia prediction score, we were able to convert the same random DNA sequence into preneuronal glia enhancer. And as you can see, after 10-15 mutations, for the Kenyon cell specific transcription factors are generated by the model. But for the preneural glia, now it was the glial specific and preneural glia specific transcription factor binding sites were generated.
Ibrahim,
And,
we
yes.
do have a question from the audience. So this is from Claire Anderson, who's asking, how would you check GFP toxicity in Kenyon cells? How do you differentiate this possibility with negative enhancer activity?
With co-staining, like we were staining with the dachshund, we are checking on the wild type flies, how the dachshund profile is, and we check with our synthetic enhancers that are positive, GFP positive, and we check again the dachshund. Okay, everything seems as wild type dachshund expressing the Kenyon cells at the original position. But then we were having some cell lines that, okay, we don't see GFP positive expression. We were thinking, oh, maybe our enhancers are not positive, they are not working. But also then if we look at the dachshund, and dachshund expression was also weird, or it wasn't there completely at all. So with this way, it was just an hypothesis, maybe the GFP toxicity, because on the literature we were saying, okay, GFP is toxic, and if this too much, if this expressed too much, it may bother the cells. And this is an hypothesis. And then what we could do, actually, we could go back to the mutational steps and choose the mutations. Maybe instead of 15 to 10 and 8, maybe it will express the GFP less. And maybe with this way, we will be able to see the Kenyon cells on their original location. We didn't do such experiments, but it's like this GFP toxicity and killing the Kenyon cells were an hypothesis. We didn't test this further. I hope this answers.
Yeah, I hope so. Thank you. I mean, I guess Claire can comment in the chat. Okay, yeah. Great.
And another way, okay, so we thought we can create an enhancer from scratch, and then we thought, okay, can we create an enhancer inside another enhancer? So there will be 2 different codes corresponds to 2 different cell types. Here, for this way, we use genomic sequences, and this one is a T-neuron enhancer, and as you can see, it is specific to T2A, T4, and T5. It's accessible there, model predicts correctly, and if you look at the activity, indeed, the enhancer is active in those T neurons. And this way, this time we choose the mutations that increases the prediction score for the gamma Kenyon cells, the mutation that increases the prediction score for gamma Kenyon cells, at the same time, doesn't destroy or increase the prediction score for the T neurons. And with this way, okay, as I was showing before for the Kenyon cells and preneural glia, there are different patterns appearing differently for 2 different cell types. Here again, on the same 500 base pair sequence space, we actually create two different codes, like 2 different patterns. And after testing in vivo, we saw that, okay, now we can augment the activity of the enhancer to another cell type in just one enhancer. And they are not using the same transcription factors. They are not using the same patterns. They are distinct patterns and distinct transcription factors, but they coexist on the same space. And the other way around, we can find such sequences that are the regions, the enhancers that are active in multiple cell types with the help of model. And we wanted to do this time, okay, we have an enhancer that is active in multiple cell type, and with the help of model, we wanted to prune them. And the way we identify again, scoring the whole genome and looking at the gamma Kenyon cell prediction score versus T1 prediction score. And as you can see, these regions, these 500 base pair sequences, they are shared between these two cell types. And we choose this one as an example. We have studied in the previous paper this enhancer. And as you can see, it's active in T neurons and Kenyon cell enhancers. Models gives a very close prediction, and it corresponds to accessibility. And when we look at the in vivo activity, indeed, it's active in the optic lobe neurons and the Kenyon cells. And this time we choose the mutations that do not bother or disrupt the Kenyon cell patterns, or doesn't create repressor for the Kenyon cells. So these are those mutations. At the same time, it destroys the T1 and T2 neuron patterns, and it creates, when we look at the network explaining techniques, these mutations were, okay, destroying the activators for T neurons, at the same time creating repressor for T neurons, and they were not creating or destroying any patterns for the Kenyon cells. And after 9 mutation, we were able to prune the activity of these enhancers to only Kenyon cells and destroying the activity in the T1 neurons and T neurons. And so far what I showed is everything we did was a single nucleotide at a time, and we were checking what's happening with each single mutation. And then every time what we observed is that these mutations, they are creating or destroying repressor or activator binding sites. And we thought if we know the needed activator binding site, then we thought, okay, we can directly implant those binding sites and we can create enhancers. We don't have to do this nucleotide by nucleotide and ask model to create a pattern with the help of these single mutations. So our second approach was motif by motif. And it's, again, quite straightforward. We start from a random sequence. We know which pattern is important for which cell type. And we implant this pattern to every possible positions. And then we score these sequences with the model. And the model says, okay, this is the best position. And then we implant this pattern to that location. And for the Kenyon cells from the previous publication, we knew that ILIS, MEF2, one-cut, and SIs, they are quite predictive for the enhancer activity. And we use those patterns to implant. And to give an example how it looks like, okay, this is the original random sequence. If I was doing single mutation one by one, these are the mutations that were going to be selected. Like this was going to be the first, I don't know, on the second, which one is going to be the top. But they are the ones, as you can see, the ones that destroys repressors in the beginning. And instead doing single mutations, if you do the motif implantation, as you can see, these are the locations that are preferred most by the model. And they, again, corresponds to a location where there are repressor binding sites. So the model wants to destroy repressor at the same time when it's creating an activator. And if it happens at the same time, then it has the most importance for the model. And another thing we noticed, like if we didn't have these deep learning models, and if we didn't know the repressor code, imagine we were implanting the ILIS binding site to this location, and actually it's surrounded by the repressors, and these binding sites were not going to be functional. And thanks to the model, we know where they are, and model helps to choose the best location. With this way, we were able to generate synthetic enhancers. And then we wanted to measure like what is the effect of each pattern. This is the random sequence. When we add SR or one cut individually, they do not increase the prediction score much. They don't have much effect. ILIS and MEF2 had the highest effect. ILIS had even more than MEF2. And when we add the ILIS and MEF2 right after each other, implant them, like first we implant the ILIS to the best location, then we take that sequence and implant the MEF2 to the best location. Their effect was greater than the sum of their individual contributions. So there's a kind of synergy in between these two transcription factors. And when we are at this level, adding one cut and SR increases the prediction score further, even though by themselves they were not functional. And now after this 4 motif implantation, we are at this level where we were with 15 single mutations. Okay, now since it's in silico, we can do this experiment 1,000 times, taking 1,000 different random sequences and implanting the combination of these transcription factors, patterns. And now, since the model put them to the best location, we can study what is the if there's any preference distance between different transcription factors. So here you can see, for example, MEF2 is in the middle and one location of one cut relative to MEF2. And usually they prefer to be in 200 base pair space, and being closer is more preferred for all these pairs. But what we observed also for the iris and MEF2 and one cut and iris, that MEF2 really likes to be 5 base pair upstream of iris. When iris here, MEF2 likes to be 5 base pair upstream. But also there are other cases where it is more 100 base pair or 200 base pair space. And for one cut, it was 3 base pair. So it likes to be at the downstream side of iris, 3 base pair away. And to see if this is something that doesn't correspond to reality, or it's something coming from the model predictions, if it is real or not, we wanted to check how they look like in vivo. And for this purpose, okay, you start from a random sequence, and maybe you are bored because I was keep saying every time I see repressors, they're by chance, and the initial iris pattern destroys this repressor binding site, and it is landed here. Then MEF2 follows 5 base pair upstream, then 3 base pair downstream, one cut. And if we check the activity of this 500 base pair in vivo, it's active in Kenyon cells, it's specific. But the only thing that we change in this 500 base pair sequence is actually putting these 3 binding sites. So we just took this part of the sequence with little bit flankings, and this is now 49 base pair sequence, and we tested it in vivo, and it was still specific and active in Kenyon cells. And with this way, we were able to create minimal enhancers with the help of model. And one last example, again, with the repressors, we wanted to test the repressors also in this case. And with single mutations, we created repressor binding sites on the flankings or in between the activator binding sites. And after creating this, the activity of the enhanced, here you can see the prediction of the enhancer. And after adding the repressors, the prediction is gone. It's 0. And if we test this in vivo, now it is not active in Kenyon cells anymore. And one interesting thing here is that, okay, this is the one cut, it is ATC-GAT. And the mutation we do here is it converts this C to G. We don't touch the activator binding site. But when we create this repressor binding site, as you can see, the model says the activator is not there anymore, even though the ATC-GAT is there. So what we notice is actually this single mutation, it creates a repressor binding site, yes, but in a way that it overlaps with an activator. So this is the repressor, this is activator. And since we don't know, it's just an hypothesis, probably the expression of repressor is higher, or the affinity of the repressor is higher than the activator. So when it binds, it is more strong, so the activator cannot bind there anymore, I guess. And with this, this is the kind of end of my presentation. We also have another approach I didn't mention. It's a generative design. Okay, this is nucleotide by nucleotide, motif by motif. And with the help of GAN models, we were able to generate sequences at once. But we were the purpose was designing enhancers, but also we wanted to learn more about the code of enhancers and how the designing step teaches us about the enhancer code. For the generative design, we couldn't observe this much because it pops the sequences at once, and we were not able to see, okay, what is the design process, what is the important part on the DNA sequence, and so on. Okay, we were using our original model to score generated sequences, and then we can see which patterns are there and not, or so on. But for us, it was less informative about the code of enhancers or how to design it more knowing what you are doing in a way. So as a summary, yes, with these kind of techniques and the deep learning models trained on DNA sequence, we were able to design enhancers from scratch. We can enhance their activity or decrease their activity. We can put 2 different codes onto the same enhancer, or if there are 2, we were able to remove them by doing kind of a surgery. Or with the help of motif implanting approach, we were able to generate very short enhancers. And with this, I'd like to thank my supervisor and mentor Stan, and also like all the people who helped me in these different projects. I was mainly a computational biologist. I'm in front of the computer. Sometimes I was helping for